I was talking to Sarah, and it's always difficult to talk to an audience that is really diverse. And I know there are people from different parts that sort of very scientific, some are really medically much further in what they do, and some are really trying to understand what stem cell based therapies are. So I'll try to do justice to all of you, so figure out if we can make a talk very smooth, that's very understandable to everybody. And I think just stop me where you think that I've gone scientifically too far into details. So that's not the idea. The idea is so the stem cells have been, in last decade or more than a decade, 15 years now, in news about, mostly about regenerative therapies, where you regenerate neurons or you regenerate muscles. But there is less talk about how they can be used for cancer, and is there a potential that they can be used for cancer. So we've done some work, and so have our colleagues over the last decade, and I'm going to talk about that. But first, I think it's also very confusing what stem cells are, and what are used for regenerative medicine, and what can be really used for cancer. So I'll give you a brief background of what stem cells are and what has really happened in the last 200 years with the stem cells in less than 5 minutes, and then I'll jump into the cancer part of the story. So we know that stem cells come from, basically the embryonic stem cells come from the inner cell mass of the embryo, and they're basically totipotent. When you hear the word totipotent and then pluripotent, it's always confusing, but let me define what these are. So when you have the stem cells in the inner cell mass of the embryo, these are potentially stem cells that can make any organ, plus the placenta and any embryonic related organs. But when they come out, when they're still in the embryo, but they become pluripotent, that's the next stage. That means they can't make placental cells, but they can make other cell types, any other cell type of the body. So this is very early in the development of an individual. So I think we will have the headings because of the projector not being aligned. So I'm going to give you the headings as well. So then what happens that all of these self-renew. And there are two, you will see what happens is that these pluripotent stem cells then form the germ layers, like the endoderm, the mesoderm, and the ectoderm. And then you get the tissue precursors, and finally the differentiated cells. So from the mesoderm come the hematopoietic stem cells and the mesenchymal stem cells, and they form skeletal muscles, smooth muscles, and urogenital system. And then from the hematopoietic stem cells, you get the blood cells, and mesenchymal stem cells, you get the connective tissue. And from the endoderm, you basically get the epidermis and the pigment cells. Right now, I think we're missing some parts here. Can we try to adjust the computer, or well, let's see how much we miss. So basically, if you look at this, so the title is the history of the stem cells. So if you look at the mid 1800s to early 1900s, cells were found as building blocks of life, and some cells could differentiate and after embryo development. In 1908, so this is in mid 1800s to early 1900s. In 1908, Alexander Maximov proposed the existence of hematopoietic stem cells, and in 1958 researchers reprogrammed adult frog cells and showed that they have embryonic potential. And 1960s Altman and Das published a paper presenting adult neurogenesis, suggesting that there are adult stem cells, and this was largely ignored at that time. So, and then 1963, I think that was the turning point where Tim and McLeod discovered maximal cells in mouse bone marrow. So that's where most of the multiple myeloma therapies come from. You actually irradiate the individual, and you take their mesenchymal stem cells from the bone marrow out and put them back into the patient. And so that was the first bone marrow transplant successfully used to treat severe immunocombined deficiency. And then 1981, first in vitro stem cell line was developed from mice, and as you all remember, Dolly the sheep was cloned in 1996 from stem cells and grown into adult healthy sheep. And I think then 2003, Sung Tao Shi discovered that children's primary teeth are a source of adult stem cells. Now, if you look at, there are lots of organs in the body that are source of adult stem cells. So if you, so what happened in 2006, and this was the turning point in stem cell development, that we always thought that when a seed becomes a fully grown tree and has a leaf, we always thought that we couldn't take the leaf and make the seed out of it, because that's how we studied and how we were raised, that okay, this is the leaf, it cannot become the seed. But what Shinya Yamanaka, who got the Nobel Prize last year for this work, what they took is they took a fully differentiated cell, a mouse fibroblast, a skin cell, for example, and they put 4 genes back, OCT4, SOX2, KLF4, and c-MYC, and they converted it into an embryonic-like stem cell. So that tells you that you can take any adult stem cell from the body that is fully differentiated, and you can make it into a stem cell. That actually sort of circumvents any ethical concerns that embryonic stem cells have, because now you don't have to go to embryos. So these are sort of basic stem cells. So you can have adult stem cells, you can have embryonic stem cells, and you can also have the induced pluripotent cells that Yamanaka discovered. So why use stem cells for cancer? So that's the title. So stem cells display intrinsic tumor-trophic properties. So that means if you have a tumor somewhere here and you have stem cells here, they sort of tend to go to the tumor. I'll explain that in detail. Can be modified to stably express, release a lot of anti-cancer agents. So that circumvents the short half-lives that many anti-cancer agents have, and then they can disseminate solid tumors and migrate towards metastatic lesions. And I'll again go into the details of all these. The next title is that what are the different types of stem cells that are used for cancer therapy? So initially people used a lot of embryonic stem cells. Again, they are primitive, undifferentiated cells derived from 5-day pre-implantation embryos. So again, we have ethical concerns regarding this. And then we have adult stem cells. These are adult undifferentiated cells found in different tissues. Like you can find stem cells in bone marrow, you can find stem cells in your teeth, you can find them in cord blood. You can find them in a number of organs, and both of them have their advantages and disadvantages. And one of the major advantages of adult stem cells is you can take it from the patient, play with them, and put them back in the same patient, so you don't have a problem of immune rejection. So the new types of stem cells are the iPS cells. That's what I discovered. These are called also the Yamanaka cells now. So these are the ones that if you have a patient and you can take patient's skin cells, convert them into a neural cell, for example, and put them back to regenerate neurons in Parkinson's disease, or if somebody has a stroke, to regenerate to cure stroke. So that's the big advantage of the iPS cells. But iPS cells are not easy to play with, as are the adult stem cells. So a lot of our work is mostly on adult cells, and people who have worked on cancer therapy using stem cells is mostly on the adult stem cells. So the question then is, where can you use stem cells, and how can you use them, and why are they so important. So all the next 30 minutes you will see slides mostly from our work here at Mass General, is why and how, and how can we take this into clinics. And one of the tumors that we chose is a brain tumor. It's the reason why we use stem cells for brain tumor therapy is 2 to 3-fold. One is that most of the drugs that reach other cancer types, if you give a systemic injection of a drug to a patient with a breast tumor or patient with a pancreatic tumor, these drugs do reach the tumor. One of the big problems with brain tumors is majority of the drugs do not reach the brain tumors because there's a blood-brain barrier. There's a barrier between the blood and the brain that doesn't allow many drugs to reach tumor. And that's very good for us also, because that's why brain doesn't get infected with viruses or bacteria. It's very immune privileged in that sense. So that's one of the reasons. And then also the tumors in the brain are a bit different than the others. So this is how the brain tumors develop. I'm talking about the highly malignant grade 4 glioblastoma. This is where patients have 12 to 18 months to live after they are diagnosed. So this is how it develops. You have some tumor cells in the brain, and you get more tumor cells, and then you have the blood vessels coming in. Again, it's one of the few tumors where it doesn't depend on the formation of blood vessels. The blood vessels come in later. In many tumors, as soon as the tumors start, the blood vessels start developing around and help the tumor to grow. In this case, they come in later. And then you get a necrotic core of the tumor. This is one of the cores that doesn't allow tumors to also respond to many therapies, because this core makes the tumor very hypoxic. There is less oxygen there, so most of the drugs do not work in such a situation. So ultimately what you have is a grade 4 full-blown tumor with blood vessels around it, necrotic core around it. So that's not the only problem with brain tumors. The other main problem with brain tumors is that some of the tumors leave the main tumor site, some of the tumor cells leave the main tumor site and go to the other parts of the brain. So you don't just get the main tumor, and this actually happens after you do the operation, and then the tumor regrows, and then you get these different micrometastatic sites, you call them in our terminology. And they also have their own blood vessels. So there's a lot of research that has happened in brain tumors over the last 3-4 decades, and people have identified different genes that are responsible for the growth of the tumors. Again, I'm not going into the details. So these are growth factors and growth factor receptors that are involved. And there are the death receptors that I'm going to go into the details, because a lot of our work is focused on targeting death receptors. And then there are also there's also angiogenesis, that is the formation of blood vessels. So basically why I'm showing this is because these also offer targets. So what we want to develop is something, some drugs that can specifically kill this tumor cell and not kill the normal cells in the brain. So for example, the death receptors are only present in these cells and not elsewhere in the brain. And similarly, some of the receptors are present only on the blood cells, blood vessels, and not in the normal brain. So you can basically target these cells, you can target these cells, and there are some of the markers like EGFR is overexpressed only in the tumor cells. So again, it's not us who is trying to develop, not only us who are trying to develop new therapies. There is an ongoing sort of effort from a lot of labs all over US and also in Europe and other parts of the world to develop therapies. But the standard therapies are that you get operation of a brain tumor patient, and then you get the radiation and chemotherapy. There are new therapies that are coming in. Again, the major problem with these therapies is that when you give them systemically, if you give an injection, an IP injection, most of them don't go to brain. And then that's where the new virus therapies are coming, and also ways how you can break the blood-brain barrier, how you can allow the drugs to go go into the brain. But let's go back to the stem cells. So our reason for developing therapies, I think, is clear. In the brain is twofold. One, we want to, because the drugs do not go to the brain, so we want something to be delivered at the site of the tumor, and then we want those drugs to be specifically killing the tumor cells and the endothelial cells and not the normal brain cells. So let's see what we have done. So first thing what we did is we looked at this is when I started here at MGH like 8-9 years ago. We looked into mice with which had a tumor, and we implanted neural stem cells on the other hemisphere of the mice brains, and in this mice there is no tumor in these mice. And we follow them over 2 weeks, and you see these stem cells tend to migrate towards the tumors, and these stem cells don't go anywhere. So at 2 weeks, a lot of these stem cells have gone into the tumor, and these stem cells haven't moved. So if you look at these mice again at the histopathology level, this is where the stem cells were implanted. This is the path they migrated through across corpus callosum, and this is how they went into the tumor. So basically it tells you that naive stem cells, unmodified stem cells, have the potential to just track tumor cells. They can seek out. So drugs normally don't do that. They just don't seek out the tumor cells. They go to every cell, but only tumor cells respond to that. So then we also color coded these. So we have now the red, the green as a tumor cell, the red as a mesenchymal stem cell. These are adult bone marrow derived human mesenchymal stem cells. And we looked at week 1 and week 2, and you see all these cells that over here have now migrated into the tumor and have gone into the tumor. Now the question then is, can we do something with these red cells? If we can modify them to release the drug, they can kill these green cells. That's the idea. Tumor cells are leaving the main tumor site and going elsewhere in the brain. These stem cells will follow them and kill them. This was our model 5 to 6 years before, and that's everybody's model before, and now 90% of the labs are doing exactly the same way. The problem is that when we did this, by the time stem cells started killing, this tumor was all over the brain. Because you have to understand that brain tumors are extremely aggressive, that you can't leave them just like there and then cure 10% and then you'll see. So then we went back and said, well, what can we do if we get a brain tumor patient? Can we take stem cells and just inject them into the brain and say, well, they're done? The answer is no. The answer is no in twofold. One is that when somebody comes to the OR and says, I have a seizure or a headache, and says, okay, let's go and do an MRI, and they find a big mass in the brain, they say, the neurosurgeons say, you got to take this out, irrespective of if it is a brain tumor or not, because the pressure of the mass in the brain will kill the person. So it needs to be taken out, and it turns out that 99% of the times this is a brain tumor. So if 99% of the times it's a brain tumor, it happens in 70 to 75% of the patients. So 75% of the patients get operation. So we thought, if we could mimic something what is in the clinics, we could have a better shot of doing things and doing them well. So we made a mouse model of brain tumor resection, where you resect the tumor, which is extremely difficult. I don't know how we still do it, but you have to understand the mouse brain is very, very small. You implant the tumor, you let the tumor grow, and then you resect like the neurosurgeons do in the OR. And then basically we saw that this is the live image. So this is the tumor in the brain, the green tumor. We color-coded green, and then one of us, I think it's one of the postdocs, removing the tumor in the brain of the mouse, and this is the tumor. This is what is remaining after the tumor has been removed, and this is the excised tumor. So we almost removed 90 to 95% of the tumor. But this is the culprit. This grows much faster than this one now. These cells that are remaining there. So this is where what happens in patients. So when you take this tumor out, and immediately after this, patients are put on chemotherapy and irradiation. And still, I think in 80 to 90% of the patients, these tumors reoccur. And I don't think we have done anything in the last 20 years to make things happen differently. Although there are a number of clinical trials out there with different drugs, but again, nothing happens. So we developed a mouse model of tumor resection where we have, this is pre-resection, this is after we resect the tumor and you remove majority of the tumor, and we can do this with MRI and with bioluminescence imaging, and we show that mice survive longer than normal. That means if you resect the tumor, mice survival is longer, but ultimately they die. So we then thought, can we do something in this cavity now? Can we develop something that is delivered right into the cavity of the mouse tumor? So we put stem cells, being very naive, and we put stem cells, and guess what? We didn't see anything because they got washed out. So you also have to understand that this cavity gets filled with cerebrospinal fluid and blood, and as soon as you put something there after the operation, it gets washed out. So, and that told us why many clinical trials before us had failed, because people who had put viruses there and had put other drugs there, they were washed out. So nothing reaches these remaining tumor cells in the brain. So sort of at this point we were lost. We said, okay, this is not going to happen because stem cells get washed out. And then we came across a company that was making biocompatible gels. So all we need stem cells here, but we need a cement. We need to paste them there, and paste them in such a way that paste doesn't actually alter our stem cells. It keeps them intact, makes them still behave the same way as they are. So we did this in the lab, in the culture dish. We encapsulated these stem cells in a biocompatible gel and then plated the tumor cells around it, and we see that these tumor cells, again, if you have trail secreting stem cells here, they kill the tumor cells. And also when you encapsulate them and implant them in the mouse brains that have a tumor, they migrate out of and follow the tumor. So the gel actually doesn't change the properties of the stem cell. It can now behave as a paste, and basically then we did the real experiment. So we have a tumor that has been removed. We put these green stem cells secreting trail in that cavity, which has still some tumor cells in there, and then we did an experiment where we looked at, if we put the cells with the gel and without the gel, you will see a major difference, that at day 7 majority of the cells without the gel were gone. So the gel helps the cells to be retained in the cavity, and what happens after that is that majority of the mice survive, because these stem cells are secreting trail and they are killing the remaining tumor cells here. So we get almost 100% survival in mice. So this study was published in Nature Neuroscience a couple of years ago, and got quite a bit of press, because it was basically simple things put into practice. There was no rocket science in there, and basically what we did is that what people could do now is put, if they have great therapeutics, stem cell based therapeutics, they can create the cavity in the mice and then in the mice tumor and put these stem cells in the gel, and again their benefits, again there's less host immune response, there's retention of cells that prevents washout. So this is great. So this is our model now. This is how we're going to go into clinical trials as well, that we're going to go into trials in a resected tumor with with stem cells encapsulated in the gel. But then there are problems. It's not. That's why the title, it says separating hope from hype. This looks great, but there are problems. Let's see what they are and how we can circumvent them. One of the things that many of us doing brain research or any kind of cancer research rely on lines that have been established like 50 years before or 30 years before, and we have a bank called ATCC somewhere in Washington that supplies this to everybody at some certain price. But now in last a decade, people have started focusing on fresh tumor samples. You take the tumor samples from patients that are cultured and you work on those because those are fresh, those are primary tumors, and we have access to, through Dr. Wakimoto here in neurosurgery, to different tumor lines that were isolated from patients, and basically these tumors behave differently than established tumors that you get from this repository. They don't form just, some form solid tumors, some from semi-solid tumors, and some do not even form solid tumors. They just go all over the brain. And I'll show you one of the examples, like GBM8 is a tumor that just doesn't form a solid mass. It just is all over the brain. So it's extremely hard to treat and resect. So that's what you see from patient samples. You see that these tumors are different, that we didn't know till 5 years before. And so understanding that is critical. And then we have also tried our stem cells in these models and shown that stem cells can track single tumor cells in the brain, which is again amazing because drugs do not actually reach every cell in the brain. And then we have developed a model with the resection model with these cells. So we try to mimic, we put a lot of cells of these invasive cells in the brain and then try to resect, and then put stem cells, the green are the stem cells, in the gel, and we again show distant migration, that they go to distant parts where wherever they see tumor cells, they seek them as far. But we could not have done this in a model that is available. It was only that we got lines from patients that told us, okay, you have this type of tumor, can you treat this type of tumor? So that's one caveat, that you need to get tumor lines, fresh tumor, primary tumor lines from the patients. The other thing is that all these tumors at some point develop resistance to drugs. And so trail is no-brainer, so you have resistance to trail as well. So 50% of the tumor lines are resistant to trail. So don't think it as trail, but think of any drug you will develop. So how do you circumvent trail resistance? How can we get trail-resistant tumors? How can we target them? So, then it's going into the biology of the tumors. And again, I'm not going into the details of these. So you can basically target different points in the signaling pathway of the that tumors rely on, and then develop therapies against those specific targets and combine it with trail. And two therapies that are exciting from our lab. One is that we have used HDAC inhibitors. These are histone deacetylase inhibitors. So in this one, you see this is a semi-resistant line, and this one you give MS275. It's a histone deacetylase inhibitor, HDAC inhibitor, and trail, and you see these cells again, all of them are killed, and these are still trying to die but can't die. And so you can combine, and we now know why you upregulate death receptors on the surface of the tumor cells, and that potentiates them to be killed. And we just showed this in vivo as well. And we have recently published another study on an alkylating agent, cisplatin, that is in clinical trials already and has been used in patients. And we show again, you get cell cycle arrest, upregulation of receptors that help trail to kill, and you get high survival in mice. So you can combine the two drugs, one that's a trail sensitizer and trail stem cells, but that's not what we want. We don't want two drugs. We want everything in one drug, because that's again giving patients two drugs, you get toxicity issues. And so what we want is just stem cells. So that's extremely difficult to do. So we want the trail and the sensitizer in one drug. So we have been working with a group in Holland who is developing EGFR. So you might have heard of antibodies, monoclonal antibodies. Again, they are so big that it's hard for them to reach the brain. So we have found, so this group in Holland has found a VHH domain of an antibody. It's called a nanobody, and we have been using these now combined with trail. I'll show you a few slides. So nanobodies have high target specificity and low toxicity, and they readily access receptor clefts. So we cloned these nanobodies, and we made them stem cells secrete these nanobodies, and we showed that they can inhibit EGFR signaling. So these are targeted against EGFR. So basically then after this we created a EGFR nanobody and trail that's now a molecule that has a sensitizer in it and trail as well. And basically what it does, it binds to both the trail receptor and the EGFR receptor simultaneously. And now if you co-culture, the red are the tumor cells, green are the stem cells, with green expressing stem cells control expressing nanobodies and expressing nanobody trail, you see majority of the cells are killed. And we also know that there's a molecular basis of this. You get caspase upregulation and cell death in trail resistant cells. And we've shown this in vivo now that mice treated with stem cells expressing nanobody trail, their tumors are almost gone, and that you get huge survival benefit in mice. So you can now make a stem cell that expresses a dual molecule. You don't have to give two therapies, and it works. So basically this is what it is. So you have a stem cell, you have a bispecific molecule that targets two different receptors, and you can kill tumor cells. And this is again published. So I'm going to have two small stories now, one on our exciting studies on oncolytic viruses. So oncolytic, and the other one on metastasis. Metastasis are the tumors, metastatic tumors are the tumors that go from one organ to the other organ. So I'll come back to that. So one story about oncolytic viruses. Oncolytic viruses only replicate in tumor cells. So that's the beauty. So they don't affect the normal cells because they can't replicate. So they infect some normal cells, but they only kill tumor cells. So when you mean replicate, when I mean replicate, that means they kill this tumor cell. So they replicate, they kill the tumor cell, and they come out, and then they infect another tumor cell, they kill it, and they come out. So as soon as they kill, they also divide themselves. So they kill the cell, but it helps them to move on and divide further. So we have a couple of years before, we have found, nobody knew this, that there are tumor cells that are also resistant to oncolytic viruses. We all thought that any tumor cell, because it's dividing, will be killed with oncolytic virus. But as nature has it, some tumor cells develop resistance against oncolytic viruses as well. And we figured out in a cohort of cells that there were two, one from patient-derived, one established line, that they were resistant to oncolytic viruses. So we had to come up with something new. So we developed an oncolytic virus now that also produces trail. So, and we showed that it targets both cell death and cell proliferation pathway. Again, I'm not going into the details of this, but if you see here, the OHSV trail kills much more significantly. Oh, well, that's missing here. You have to trust me on this one. Kills much more significantly than just oncolytic herpes virus. And then the model again is true. Now, if you look at oncolytic herpes virus, again, we are not the people who have started all this oncolytic herpes virus. There are a few groups here at MGH, Sam Rapkin's group and Bob Matuza's group. They've done some amazing work on oncolytic viruses and have taken some of it into clinical trials. And what has been done in clinical trials is that you inject the virus in this cavity, but majority of them again have failed. And as I told you in the beginning, why? It gets washed out. So it doesn't make sense to put viruses in this cavity, because it doesn't do anything on the cell. So we thought, okay, let's put viruses in stem cells. It was a very naive idea, but because we knew that stem cells would also be killed. Because stem cells divide in culture, but once they go into the mouse, they don't divide. In the culture, we give them growth factors, so they divide. So we put stem cells, these viruses, they are red colored, and we put them in the stem cells, and we see that after 120 hours, I don't know how many days this is for, 5 days? Yeah, after 5 days, stem cells get killed. Now, if you have a tumor and you co-culture the stem cells with the tumor cells, you again see that the stem cells get killed, but it also kills the tumor cells. So this is explained here beautifully. So this is one of my favorite slides. So you have, this is the tumor in the brain, the green, and we come with stem cells that we load 6 hours before implanting into the mice brains. So we load them with the virus that's red colored, and we inject them directly into the tumor. This is not a resected tumor, this is an intact tumor, just to understand what happens. So this, at 24 hours, you see the red cells are producing virus, and that is going into the green cells. When they become yellow, that means the virus has gone into the green cells. So at 48 hours, what happens? More green cells become red. That means the virus has entered the green cells more and more. And at 72 hours, more green cells become red. And again, this is the high mag of this. And then these are the details. So at 120 hours, majority of the cells are either red or dead. So there are no green cells. So basically, the virus has come out from the stem cells, it has gone into the tumor cells, and tumor cells have helped the virus to kill further tumor cells. So this is much better than the protein therapy, but we also have trail in it, so it also helps to kill further. So now we try to translate this into a resection model. So we took the virus with the firefly luciferase, that's a plain virus, and this is stem cell loaded virus, and we put it in the resection cavity, and you see the virus in the beginning, you see it, and after day 5 it's gone. So here what happens, you see the virus, but then the virus transfers into the tumor cells, and you see then the tumor cell, the signal coming from the tumor cell. And you see it here as well, that the stem cell, the virus not loaded with stem cells is gone, and the virus with stem cells is still intact. And you see in these mice, you see a huge survival difference. These mice implanted with stem cell loaded virus survive longer. And this is again got a lot of press this year, that you can now use stem cells with herpes virus to kill tumor cells in the brain. So and then, can you make these safe? One of the questions that FDA has for us is, okay, it's great to have stem cells, but how safe are these stem cells? So we have sort of incorporated a HSV TK, is called herpes simplex virus thymidine kinase, and it allows it to selectively sensitize and TK expressing cells. So once the stem cells have released trail, you can now eradicate the stem cells with ganciclovir, because these stem cells are also engineered with HSV TK. The beauty of the HSV TK is it also allows us to image the stem cell fate by PET. Now, if you have bimodal stem cells that have both trail and TK, they have the same killing potential as trail, but they can be eradicated as well at the end after the therapy is done. So that's the safety component of the stem cells. And the last part is the metastasis. So 3 slides more, and then we'll have a little question and answer. So we also know that around more than 50% of melanomas, people who have had melanomas, get relapse as metastatic tumors in the brain. 35% of the breast tumor patients who have had breast tumor get relapse in the brain. So these are now breast tumors, lung tumors, and metastatic melanoma tumors that go to the brain. So we're also in parallel working on curing those tumors. And we've recently, so this is what happens. For example, brain metastasis is very common. with lung cancer, breast cancer, and melanoma. Around 20 to 40% patients with breast cancer develop brain metastasis. So it's not only at one spot, you get multiple spots. And you see this in the MRI, you get parenchymal brain tissue and you get leptomeningeal linings of the brain. They are at different parts in the brain. We've developed a mouse model that mimics exactly what happens in the clinic. So we inject the tumor cells, metastatic tumor cells, in the intracarotid artery, and you can now image these. It actually is similar to what you see in the clinics. And we've now injected stem cells in the intracarotid artery that are releasing trail and have shown that they specifically target these tumors in the brain, these metastatic deposits, and can kill them by inducing cell death. And you see significant differences in the tumor volumes in these mice. So basically, you can now, we also have, I'm not going into the details of that, we also have melanoma model that we can inject the stem cells in the intracarotid artery, because we know that metastatic tumors, most of them don't get operated on because it's too late in the game and they're all over the brain. So it's better to develop therapies that you can inject systemically, and we know if you inject in the artery here, intracarotid artery, the stem cells go to the brain, seek the different tumor cells, and then kill the tumor cells. So basically you can have stem cells as drug factories, and we believe that developing tumor models is critical to understanding. So if you don't develop tumor models that mimic clinics, I mean, say the research is not useful at all. You have to understand what is going between the clinics and the lab. You can't be totally not in connection. And then imaging is a great tool to help provide us key information about stem cells and the treatment efficacy. And I think, as I said, that there's a big hype about stem cells, and not particularly about cancer, stem cell therapies for cancer. So it's important that we should not that we should have greater biological understanding. In my talk I didn't say, but we put a lot of effort on understanding why the tumors don't get killed, why they get killed, and what can we do to make majority of the cell population kill. But also there is a lot of preclinical fervor that should be tempered with caution, because we just can't say, oh, this is great therapy, let's go to the clinics. I mean, we are now having talks with the FDA, and we are in the pre-IND phase to take this into clinical settings. But there's lots of questions. What stem cell to be used? What stem cell would you use? Would you take a stem cell from the patient? Ideally, we would like to take it from the patient, but then engineer it and put them back in the patient. But then the timing between a patient knows that he or she has a brain tumor and has to be operated is very small. And then you can't take stem cells from them and modify. That will take months and put them back. So you have to understand the caveats. And so one of the things that we come up with is having an allogenic stem cell that is already in clinical trials in stroke patients. So we have that, and we're taking that into the patients. It has less immune rejection, and if we can put this in the and the second thing is that most of the clinical trials in brain tumor patients are in recurrent tumors. That tumors that reoccur after 4 months or 5 months, it's too late. I mean, there's no point in doing a clinical trial where we know that patient has no hope. So we are pushing FDA also to have clinical trials right at the site, right when we do the primary resection at the first attempt, because there's no point going further. And this is where we have lost the battle in this game, because we have been just doing the same thing as with other tumors, and we have to understand that tumors are different. Brain tumor is a completely different entity than other tumors. So you can't have the same things for other tumors, and we're trying our best to push FDA to see outside the box. Hopefully they will. So there's again, I think I discussed all.